Hello everyone, today I'm going to show how to add all points of glycolysis into one diagram. And you can see I have written the pathway with its enzymes and there are some pencil written things, and I'll show how to add them to the pathway step by step. So let's start with marking the rate limiting steps. The first step, glucose to glucose 6-phosphate, is a rate limiting step of glycolysis. Okay, and the next step is fructose 6-phosphate into fructose 1,6-bisphosphate. Okay, so it is also a rate limiting step. And the next step is TEP into pyruvate. So next we have to do is marking the number of carbon atoms of each molecule. Okay, then you know that glucose is a hexose sugar, so it contains 6 carbon atoms, and I have to tell that the kinases, isomerases, dehydrogenases, and mutases and enolases, they do not change the number of carbon atoms in the reaction. Okay, so here is also C6 because there is a hexokinase, and isomerase does not change the number of carbon atoms, so this also C6, and fructose bisphosphate 1,6-bisphosphate C6, because here is a kinase, it does not change the number of carbon atoms. Okay, and here fructose 1,6-bisphosphate give two molecules which contain 3 carbon atoms. Okay, but dihydroxyacetone phosphate isomerized into glyceraldehyde 3-phosphate. Okay, so we can get 2 glyceraldehyde 3-phosphates to continue their pathway. So here it can get 2. Okay, so this is a C3 molecule, and this is also a C3 one, and it become 1,3-bisphosphoglycerate by the action of a dehydrogenase, so it does not change the number of carbon atoms. But you have to remember here there was 2 atoms, 2 molecules of glyceraldehyde 3-phosphates, and here also should be 2 1,3-bisphosphoglycerates. And I told that kinases, mutases, and enolases do not change the number of carbon atoms, so we can put C3. Also here you can get C3 and C3. So and you know that there was 2, and here also 2, and here we get 2, here also 2, and you can get 2 pyruvate. Okay, do you know that for the action of the kinases, mutases, and enolases, they require magnesium ions, so we can put magnesium ions for kinases. It is important because sometimes they give MCQs what requires magnesium ions for their action. So mutases also need magnesium ions. And finally the kinase, pyruvate kinase, also need magnesium ion. Okay, Then I'm going to see the ATP and NADH production in glycolysis. Okay, so follow me. The first step, utilize the ATP. So ATP to ADP. So I write here minus ATP for this step, and phosphofructokinase step, and it also utilizes ATP. Okay, so I put minus ATP here to this step, and you can see this is the only step that produces NADH in the glycolysis. Okay, so NAD plus 2 NADH. Okay, now you saw that there are two. Here also two. So this NADH also there are two NADH produced. So I put plus 2 NADH for this step for the calculations. And there is a step that produces ATP. Okay, good. ADP to ATP. So I put plus 2 ATP. And here, finally, ADP to ATP. You know that substrate level phosphorylation. So here are 2, so 2 ATP. Put plus 2 ATP. So it is completed now, and let's calculate. So I figured that when the conditions are anaerobic, pyruvate is converted to lactate. It utilizes NADPH. So we can write NADH to NAD plus. But here are 2 pyruvate molecules and 2 lactate is produced, so 2 NADH is utilized. So I put minus 2 NADH. So let's calculate. Here you can minus 1 ATP. So there's 2 ATP and 2 ATP. So in aerobic conditions, we can put 2 ATPs are produced. Net production. Okay. So and then product of anaerobic condition. Aerobic condition is pyruvate, and 2. And aerobic condition is lactate, 2 lactate. And here we can get 2 NADH molecules. So I write here 2 NADH, and so, but in aerobic condition, the pyruvate is converted to lactate and NADH are utilized. So no net production of NADH in anaerobic conditions, but ATP remains no effect for ATP in anaerobic condition. Okay, so that's the calculation, and now I'm going to add some additional steps that are related to glycolysis. Okay, so glycogen, you know that glycogen, it can participate to the step glycolysis as this: glycogen into glucose 1-phosphate like this. Okay, and there is some more steps. Here you can see that fructose 6-phosphate can be converted to fructose 2,6-bisphosphate by the action of a bifunctional enzyme. Okay, and this fructose 2,6-bisphosphate is important, I tell you. Okay, and next additional step can be found only in RBCs. You can see here the 1,3-bisphosphoglycerate can be converted to 2,3-bisphosphoglycerate, 2,3-BPG. I hope you are familiar with it, because physiology we call the affinity of oxygen to our hemoglobin. When we discuss about it, we frequently tell about the 2,3-BPG. Yes, and the oxygen association dissociation curve. Yeah, I know you know that. And 2,3-BPG can again converted to 3-phosphoglycerate by the action of this enzyme 2,3-B. Okay, so that's the additional steps of glycolysis. And then now I'm going to talk the most important part, the regulators of the enzymes. Okay, so let's look at the first step, the rate limiting steps, I mean regulators. So I use blue and red colors. Blue for which induces the action, and red for which inhibits the action. Okay, so in liver and pancreas we can find glucokinase enzyme which catalyze the first step. Okay, so insulin come and induce the action, and glucagon it come and inhibit. So you know that in diabetes mellitus patient the insulin glucagon ratio is decreased, so the predominant action in the body goes to the glucagon. So these rate limiting steps are inhibited and the rate of glycolysis is inhibited. Okay, now there is something called GKRP. Okay, yes, GKRP. Okay, glucokinase regulatory protein in nucleus of the cell regulates the activity of glucokinase through reversible binding. Okay, so you know that here the fructose 6-phosphate it comes and tightly bind with this GKRP. Okay, and so the release of glucokinase does not occur. But when the glucose levels are high, okay, GKRP releases glucokinase. So I put plus mark for this and minus mark for this. Okay, then except the liver and pancreas, all other cells, in all other cells this step is catalyzed by hexokinase enzyme. So when there is an inhibition of phosphofructokinase 1 enzyme, hexokinase is also inhibited. Okay, inhibition of PFK1. You know that here is the PFK1 enzyme, no? And hexokinase also subjected to the end product inhibition. The end product of this reaction is glucose 6-phosphate. Okay, so it is inhibited. And but we have to remember that MCQs are asking that glucokinase, that it is not subjected to the end product inhibition. Okay, so keep it in mind. So next we are going to talk about the phosphofructokinase, the next rate limiting step of the glycolysis. Okay, so as we saw early, the insulin comes and induce the action, and glucagon, it's very bad, and it's minus. And when there is a depletion in energy sources of the cell, it means the MP is high in cells. Depletion of energy sources of the cell. So it increases the activity of phosphofructokinase to produce, to go through the pathway and produce ATP. Okay, and when there is high abundance, high energy compounds, phosphofructokinase is inhibited. That means ATP, when there is a lot of ATP. Okay, and the most potent inhibitor is activator is fructose 1,6,2,6 bisphosphate. Okay, so because this acts as an intercellular signal indicating the abundance of glucose. Okay, in fed state, the levels of fructose 2,6-phosphate increases. Thus, the rate of glycolysis is also increases. But in fasting, the concentration get decreased. This concentration, okay, and resulting inhibition of glycolysis. And there is a substance called citrate. What is citrate? It is an intermediate of TCA cycle. It also causes for the inhibition of phosphofructokinase 1 enzyme. And then let's go to the last rate limiting step. Pyruvate kinase. So it's easy. The insulin glucagon come and phosphorylate the enzyme and inactivate it. And insulin come and activates, increase activity. And you have to see that increase activity of phosphofructokinase. Phosphofructokinase activity, when there is increased activity of phosphofructokinase, it elevates the fructose 1,6-bisphosphate levels in the cell. Okay, so it which in turn activates down, going down, activates pyruvate kinase enzyme. Okay, so so that's the thing about the regulators of enzymes. So now I'm going to talk about the other inhibitors of the glycolysis pathway. Okay, so we get here one inhibitor which iodoacetate, which irreversibly inhibit the glyceraldehyde 3-phosphate dehydrogenase enzyme, iodoacetate. And here we can get which inhibits the enolase enzyme. There are the fluoride ions. So how it do it? Fluoride ions inhibit the enolase enzyme by forming a complex with phosphate and magnesium ions in active side of the enzyme, blocking the access of the substrate. Yes. Okay, and this stops entire glycolysis pathway. Okay, and the uses of this fluoride ions, there are, yes. The sodium fluoride is added to the samples of blood collected for glucose estimations, and the fluoridation of water decreases dental caries because it reduces the lactate production by the mouth bacteria. Okay, so this inhibits, okay, stops entire glycolytic pathway. And there is another compound. You know the arsenic poisoning. So in arsenic poisoning, what happens? The arsenic compete with the phosphate forming one arseno three phosphor glycerate. Okay, and arsenate, not arsenite. Okay, remember that arsenate is in another place. Okay, it is frequently asked in question. Okay, and this complex spontaneously hydrolyzes into a 3-phosphoglycerate, but we can see that here is NADH is produced and ATP is produced, so now there is no NADH produced, no ATP is produced here. Okay, and so this does not inhibit the glycolysis pathway, but can prevent net ATP and NADH production. Okay, so the last thing I want to discuss with you is the deficiency of pyruvate kinase. Okay, so when there is a deficiency of pyruvate kinase, we can see a hemolytic anemia. So why hemolytic anemia? I'll explain it. Yes, because the glycolysis is the sole energy source for the RBCs. Okay, and in this condition the rate of glycolysis is decreased. Why? Because this step is a rate limiting step of glycolysis. So when there's a deficiency, the rate of this step is decreased and the rate of glycolysis is decreased, and what happens? The rate of ATP synthesis is decreased. Then red cell membranes can't maintain their integrity, and it causes premature death of RBCs and lysis of RBCs. So we can say that there is occurrence of hemolytic anemia. So that are the important points of glycolysis, and I think you is important for you to get all the points to a one diagram, and when there is an exam you can quickly look and get an idea about the pathway. Okay, so that's all for this video, and I hope you it will be useful for you, and thank you for watching. Let's meet from another video.